Welcome to week 7, general biology at Rogers State University. This week we're starting chapter 3. Last week you finished your exam over the first several chapters of the semester. That was unit 1. Now we're starting unit 2. The first chapter in unit 2 is chapter 3. This is cells, everything you want to know about cells, everything we need to know about cells. Remember, cells are the basic unit of all living things. We can make a pretty emphatic statement that all organisms, all living things, are composed of cells. It's pretty scientific. It's a very emphatic statement. It's the smallest unit that all life can be limited to and still function independently. And one cell, even one cell, it maintains all of the basic characteristics of living things. Now, the actual cells are microscopic. They're very small. And we didn't know about cells until they were actually discovered under a microscope. Anton von Leeuwenhoek was a Dutch trader. His father was a trader. He was a lens maker. He made lenses for glasses. And he actually developed a lens that would magnify small things much larger, almost more even than just a plain magnifying glass. Here's his very crude microscope here. Hey, some of the things that he saw were, for instance, one of the first things he saw was sperm cells of all things, and so he began to see things microscopically, and you can imagine what that did to the scientific world. Now we know of microscopic living things, and that's what the invention of the microscope did. So a few years later, some scientists in the mid 1800s, they developed a theory about cells. And and again, it didn't become obvious until after the invention of the microscope. Here's the main thoughts about this theory about cells, what we call the cell theory. First of all, number one, all organisms are comprised of one or more cells. They're either unicellular, uni meaning one, or they are multicellular, meaning more than one cell. The second part of this theory said that the cell is the fundamental unit of all living organisms. In other words, all living things are comprised of cells. And the third component of that theory was that all cells come from pre-existing cells. I know you and I, that sounds rather redundant, but we're talking the 1800s, not too far removed from the Dark Ages. They felt like life could generate spontaneously from just about anything. But scientists began to doubt this with the invention of the microscope. Now they said, no, cells come from other cells. They are derived from pre-existing cells. In order to have cells, you had to have cells that existed before that. Those are the primary components of what we call the cell theory. And there's some other things we could talk about. Cells use energy, cells have DNA and things like that. But those first three there, the primary components of the cell theory. All living things are comprised of cells. Now, why don't we see big unicellular organisms wandering around our planet or around town? Or, why do we have humans that are comprised of trillions of cells? Why don't we just have large one-celled organisms like the blob? Well, there are some reasons for that. Here's two cells here. One is the eukaryotic cell on the right. You and I are comprised of eukaryotic cells. We've talked about that. Bacterial cells are comprised of prokaryotic cells, much smaller. Look at the discrepancy in size. Bacterial cells are so small, you could fit thousands of them inside of one eukaryotic cell. So cells vary greatly in size, but why are they so small? And prokaryotic cells, they don't have a nucleus. In fact, they don't have any really structural things inside the cell other than some DNA and ribosomes and things, but eukaryotic cells are much more complex and larger. Well, here's one of the reasons why cells are so small. And it's a difficult dynamic to kind of understand, but kind of stay with me. And we have to think three-dimensionally here, not just flat dimensions, but three-dimensional. If we have a small cell surrounded by a membrane, think of a balloon that's just barely got a little bit of air in it, and it has a membrane that surrounds it. Well, everything that wants to enter the cell or leave the cell has to pass through that balloon or that membrane. If we blow that balloon up more and even bigger, you can see that that membrane that surrounds it, the surface area of that membrane or the latex of a balloon, does not keep up with the volume of the internal part of the cell. The bigger the cell gets, the internal volume grows at a faster rate than the membrane around it, or in this case, a balloon. And so that really limits the size of a cell. There are mechanisms that only allow certain things in and out of the cell, and they must pass through that membrane or the latex layer of a balloon. The bigger the volume of the cell, the harder it is for everything to pass through that membrane that needs to pass through. So the surface area of a cell limits the size of a cell. And also the second thing is the nucleus controls how large a cell gets. When the internal volume of a cell reaches a certain size, there's a triggering mechanism inside the nucleus. And once that trigger is activated, the cell goes through a division process. Now think about that for just a second. I want you to let you ponder that. If we could control that mechanism, if we could control that triggering mechanism, think of the diseases that we could solve or get a better understanding of or cure. Cancer is an abnormal rate of cell growth. Most cancers are abnormal cell growth, that accelerated cell growths. If we knew what that mechanism was and if we could interrupt it, think of the diseases we could cure. Well, those are really the two things that control cell size. The membrane, the surface area of the membrane compared to the volume of the inside of the cell, and a threshold triggering mechanism of the nucleus that once it is activated, the cell divides into two smaller cells, what we call cell division. Now, we can kill cells and we can kill cancer cells. We can take chemicals or use ultraviolet light or radiation to kill them, to stop them from growing, but there's some, as you would suspect, some pretty serious side effects to that because we're killing good cells as well. And so that mechanism is not easy to interrupt. That's what a lot of vaccines do, is we try to interrupt. In fact, that's what the COVID vaccine was. Let's see if we could somehow interrupt the replication of a virus, and that's where most vaccines kind of center on. So let's talk a little bit more about cells. Remember, we've talked about prokaryotic cells. They are very small, very ancient forms of life. They're very simple in structure. They don't even lack a nucleus that houses their DNA. Bacteria are prokaryotic cells. Archaea are prokaryotic cells. In fact, 2 of the 3 domains are prokaryotic cells. And here's some pictures of them here. And these are really, really blown up here, and accentuated with color just for contrast. But prokaryotic cells, number 1 here, very small, very primitive. They lack internal structures. They have DNA, but they lack most internal structures. Then here's number 2, eukaryotic cells. We've talked about this. They are larger. much more complex. They have many internal structures. They have a nucleus. They have what we call organelles that are also surrounded by membranes. For instance, here's the nucleus surrounded by a membrane of this cell here. Look, here's a plant cell. Here's some chloroplasts, green chloroplasts that are surrounded by a membrane. We call these organelles. They're not organs. They're mini organs, M-I-N-I organs, called organelles. And here's the examples of prokaryotic cells: protists, protozoans, amoeba, paramecium. All fungi are eukaryotic cells. Plants and animals are eukaryotic cells. Much more complex structurally. And this is what we will spend most of our time on. But let's talk real quick, just a little bit about the bacteria, the archaea, and some of the cellular features that are common to all three domains. Well, all three domains are comprised of cells. They all have cells, and they all are surrounded by a membrane, what we call a cell membrane. There's only one domain that has where the cells have a nucleus, also surrounded by a membrane, and that's the eukarya. But here's a nice chart kind of distinguishing the difference between prokaryotic and eukaryotic cells. Bacteria, which are prokaryotic cells, they lack any internal structures that are surrounded by a membrane. They do have some ribosomes that are used in building proteins. Their DNA here is just floating around inside the cell. It's not bound inside of a nucleus. Look here, here's the membrane. This cell membrane surrounds every known living cell, whether prokaryotic or eukaryotic. And then there are some other things. Here's some material outside of many bacteria called the capsule, and that kind of protects it. Here's most all bacteria, a majority of bacteria have flagella that are used for movement, just like human sperm cells use flagella for movement. But that's the anatomy of a bacterial cell. Pretty simple anatomy. But look, this is an animal cell, much larger, much more complex. Look here, eukaryotic cells also surrounded by a membrane. This is a nice three-dimensional view of an animal cell surrounded by a membrane. But look inside. Here's a nucleus surrounded by a membrane. Here's some structures surrounded by their own membrane. Here's some other structures surrounded by their own membrane. See, prokaryotic cells lack these complex internal structures, but eukaryotic cells have them. And this is an animal cell here. And so membrane surrounding the cell and membrane surrounding each individual structure. Here's a plant cell. It's green. Most plant cells are green, not all, but some plant cells are green. Again, surrounded by a membrane. Now, plant cells also have a structure here outside the membrane called the cell wall. They also have all of these membrane-bound structures inside the cell. Here's a chloroplast, here's a mitochondria, here's the nucleus. So that's indicative of eukaryotic cells. Complex, surrounded by a membrane, and they have structures inside also surrounded by a membrane. We call those organelles. Well, here's this membrane that we've been talking about that surrounds the cell. This is kind of a three-dimensional view, but here's a plant cell with a membrane that surrounds it. Every cell, prokaryotic, eukaryotic, primitive, complex, every cell is surrounded by a membrane. Think of that balloon that's blown up just a little bit, that latex area around that balloon, three-dimensionally. That's the cell membrane. Extremely important. We're going to talk for the next several slides here, about 5 slides, about this structure, and then we'll take a break. But this membrane, this cell membrane, it's the barrier between the internal cell and the external environment. It forms a barrier. It's a protective barrier. It protects the inside of the cell from outside. So it's extremely protective. Also, look at this, it regulates anything that wants to go into or out of the cell. It maintains homeostasis, a constant environment. So those are the two primary functions of a membrane around every cell. It forms a barrier between the interior of the cell and the exterior environment, and it regulates the passage of materials in and out of a cell. Think about this. Every cell has to have oxygen to function. Every cell has to get rid of carbon dioxide to function, just like you do. Every cell has to bring in glucose for energy. Many cells make things that are excreted to the outside: tears, saliva, mucus. Many cells produce substances that are for the greater good of the overall organism. And so many cells build things inside and then excrete them outside. Many things move into the cell. So I'm beating this to death here, but you understand what I'm saying. And the membrane is what regulates the movement of substances inside and outside the cell. Here's how the membrane is constructed. It's really extremely simple for such a complex organ in cells, but here's a fat molecule. These are fatty acids. We've talked about this in the last chapter, chapter 2. Fatty acids. Here's a glycerol molecule and a phosphate molecule. This is what we call a phospholipid. It's a lipid, a fat, with a phosphorus molecule attached to it. We call these phospholipids. Now, here's what it looks like under the microscope. This is one phospholipid molecule. Here's the phosphate end of the molecule, and here's those fatty acids here. And that's what comprises the membrane around a cell and the membranes of everything inside the cell too, because we said they are surrounded by membranes. Well, think about this for a second. This part here, the phospholipid area, is what we call hydrophilic. It is attracted to water. But these lipids, fats, you know this, oil and water don't mix. This is what we call the hydrophobic part of this molecule. So this molecule has a portion that is attracted to water and a portion that moves away from water. It's phobic. Hydrophilic versus hydrophobic. Cells are almost always in an aqueous solution. By aqueous, we mean water environment. They're almost always in, even the cells in your body, they're in much of your body is water. So they're in a very aqueous environment. So when you take trillions upon trillions and trillions of these molecules and place them in an aqueous environment, something really cool happens here. So we have a water interior of a cell and a water exterior of a cell. So when we have trillions of these phospholipid molecules, one end of the molecule is attracted to water, one end of the molecule is repelled by water. That's how membranes form. We call it a phospholipid bilayer. Out here are the hydrophilic parts of these molecules. In here are the hydrophobic parts of these molecules. So literally the polarization, attraction, and repulsion of these molecules to water is what forms this membrane. They're kind of bonded together by, oppositely charged negative versus positives, and that's what forms this layer around every cell and every structure inside the cell. It's what we call a phospholipid bilayer. There are also a lot of proteins in this phospholipid bilayer. These proteins are embedded. Sometimes they extend all the way through the membrane. Sometimes they form channels in the membrane. Sometimes they're just simply on the outside or inside of the membrane. These proteins have a lot of different functions. Some are transport proteins. They allow substances in and out. This is a channel protein. Things like water, especially, can just usually freely move through these channel proteins. Enzymes oftentimes move through here. But some things are large that need to enter or leave the cell. It's the proteins that move substances in and out of this membrane. Now, this is the last slide, next to the last slide that we're going to talk about, but I need you to think about something here. Here's a very important membrane right here. Protein. It's on the outside of the cell membrane. Here's a cell membrane. Here's the interior of the cell. Here's the exterior of a cell. That protein right there is extremely important. It's the fingerprint of this particular cell. It has a very specific identity of what kind of cell it is, where it's located, and the components that want to go in or out. You see, we don't want substances just freely moving in and out of cells. If it's going to harm the interior of the cell, we don't want it in your cell. Think about the brain. There's a very specific layer of cells called the blood-brain barrier, BBB. It's a barrier between your blood and your brain cells. Not everything that's in your bloodstream do we want attaching to your brain cells. Chemicals, things like that. So we form a blood-brain barrier. Well, these proteins, especially on the outside layer of the cell, are what determine gets into or out of the cell. That word determine is very important. Usually it's a polarity, it's a chemical identity, but nothing gets into the cell unless that protein recognizes it. Now, I need you to think about something. What makes you different from another person in your household or in your apartment or at your work, wherever you're listening to this lecture? What makes you different? Well, I know you're thinking big scale, personality different. No, I'm talking even more specific. Hey, let's talk about organ transplants. If you needed a kidney or whatever, you need a kidney from somebody who's closely related to you. You know why? Because these proteins right here are unique to you and you only. You have fingerprints. This is the fingerprint of the cell. This is you. We call this self. These proteins on these cell membranes identify self. Anything that's not self, this cell considers it an enemy, non-self. And your body will do anything it has to do to attack it, kill it, or get it out of the system. That's how important these proteins are. They are what we call recognition proteins. Some are receptor proteins. Something only gets into the cell if it's received by these proteins. So you see the importance of these proteins in this membrane. Extremely, extremely important. Last thing I want to talk about is there's some cholesterol embedded in this membrane, steroids, cholesterol, and usually it gives it some rigidity. gives it some structure and support. So really the primary components of the membrane are the phospholipids here. One end is hydrophilic, one end is hydrophobic. There are proteins embedded in this cell membrane, also on the outside of the membrane. And then there's cholesterol embedded here for structure and support. So what we're going to do in the next part of chapter 3 is begin to talk about all of these things inside the cell, inside the cell membrane. Let's talk about their functions, a little bit about their structure, but most importantly, their functions. Most of these you heard of in high school biology, so we'll do that in the next section of notes.